The overall goal of combining artificial microRNA-mediated RNA interference with optogenetics is to characterize the effect of gene knockdown on presynaptic function within intact neuronal circuits.
This method can help answer key questions in the field of synaptic physiology, such as addressing the physiological role of presynaptic proteins in intact brain circuits.
The main advantage of this technique is that it makes it possible to monitor synaptic transmission selectively at neurons with reduced expression of the presynaptic protein under investigation.
Although we have used this method to investigate presynaptic function in acute brain slices, it can also be applied to manipulate and probe neuronal security in vivo.
Demonstrating the procedure will be Carmela Vitale, a grad student from our laboratory.
After designing the microRNAs and constructing a recombinant vector to express the microRNAs and the optogenetic probe, prepare primary neuronal cultures from the brain region of interest. To do this, follow an established cortical culturing protocol by L.A. Cingolani and company with the following modifications. First, use 6 well plates, plate 500,000 neurons per well, and use 2.5 milliliters of attachment medium per well. After incubating the plate for 4 hours, switch to 3.3 milliliters of maintenance medium per well, and if astrocyte overgrowth is observed, add 0.5 milliliters of maintenance medium, supplemented with 7.5 micromolar of Ara-C at 3 to 4 DIVs.